Okay.
Hello, my name is Ivy Tang. I'm currently working in the Chem Lab under the supervision of Dr. John MacArthur in the Department of Chemistry. My project is the biocatalyst discovery for the synthesis of lacto and tetraose. So there are over 200 known human milk oligosaccharides, but many of these cannot be individually characterized because we currently can't synthesize them. Although we can't individually characterize them, we do know that as a whole, they're involved in neurogenesis and immune system development. Also, they're less abundant in cow milk, as you can see by the figures. Cow milk consists less than 0.1% of oligosaccharides, whereas human milk consists of 8% oligosaccharides. So because infant formula is made from cow's milk, we don't have a lot of oligosaccharides in baby formula. So we would like to synthesize these oligosaccharides to figure out what their biological activities are. To synthesize these, we need one glycosyl transferase per linkage between the sugars. And these glycosyl transferases, they must be highly active and well expressed in E. coli, so we can actually synthesize them. And for some linkages between sugars, no suitable enzyme has been discovered, so we can't create some of the compounds we want to make. So for example, up here next to the X, that is the link gal-beta-1,3 glutamate. So we currently can't make that. So all the circles over there, it means that we also can't make those compounds. And that includes LNP, which is part of the project that I'm part of. And since these are core structures, it means that we also can't create larger human milk oligosaccharides because larger human milk oligosaccharides need these core structures inside them. We also can't make derivatives of these core structures because to make derivatives, we need the original. So here are some examples of LNT derivatives in human milk. Specifically, here's LNT over here, and then here's DS-LNT over here. So DS-LNT is interesting because past research has shown that DS-LNT can protect neonatal rats from necrotizing enterocolitis, which is tissue death in the intestine. So maybe if we can create LNT, we can add it to baby formula and protect infants with immature immune systems from necrotizing enterocolitis as well. So for our research, we would like to discover a beta-1,3 galactosyltransferase for the preparation of LNT. After we make this, we would like to optimize the reactions for high yield at a low cost production of LNT, maintaining the pH and temperature of the conditions. We would like to have a high yield and low cost so that we can add it to baby formula, so it's at a reasonable price for consumers. So how do we choose these enzymes? We currently know that WVE-1 from E. coli and HT-beta-3-gal-T from H. pylori have low expression yields in E. coli, but since they express, we can possibly use them in optimized conditions. Also, we got 4 homologs of each of them by using BLAST from the NCBI database. We ordered these genes using Invitrogen. So our question for my project is, do any of these enzymes display sufficiently high expression level stability and high levels of the desired catalytic activity for the affordable and scalable LNT synthesis? So how did I do this? Well, first I start off with DNA cloning. So we did polymerase chain reaction, which amplifies small amounts of DNA. After this, we mini-prepped our cell culture to obtain PET22B+. And this is an E. coli expression vector. After this, we digested our DNA by using endonucleases SAL1 and NDE1. And this prepared to be ligated so we can ligate our sample into the vector. After this, we transformed our DNA into E. coli DH5 alpha. We specifically chose this strain because this strain allows for a higher transformation yield. So these are the results for our polymerase chain reaction. In the table shown, you can see our temperatures and the time and how many cycles we did for each. During this, we denatured, annealed, and extended our DNA, and we stored it at 4 degrees Celsius. The picture over here is a gel. You can see that we used a 1 kilobase ladder, and the red over here is our bands. So you can see that they're all very concentrated, which shows that our PCR was successful. So for the next parts, we transformed our colonies, but they had empty vectors. So basically, our desired gene wasn't in there. This normally indicates that there was incomplete digestion of the vector, but since this pollen's been going on throughout the whole lab for about a month now, we realized that it was probably a contamination of the mini-prep solution. So we did this experiment where we had a 1 kilobase ladder over here, and then this was a mini-prep using the old solutions, and then this one was a mini-prep using the new solutions. So we're supposed to have one concentrated band, but as you can see in the old solutions, it's streaked, which showed that there was contamination. And we later found that our resuspension buffer was contaminated. So we replaced it, and now they're working better. So after this, we would digest and ligate our samples, and we're going to soon transform our DNA into DH5 alpha E. coli. So after we transform them, we'll take the colonies into a culture, and we'll induce protein expression by adding an IPTG. After this, we'll purify our proteins through visual affinity chromatography, and we'll check to see how pure they are by using an SDS-PH gel. After seeing which proteins are pure, we'll put them through dialysis so we can remove all the unwanted buffers. And then finally, we'll use a Bradford assay to see how concentrated they are. The more concentrated the proteins, the better, because then it shows us that our enzymes have a high expression yield. So after we find a good enzyme, we'll react it with various nucleotide sugars to see its properties, and then we can also figure out the kinetic constants of these reactions. We can then figure out the optimal pH and temperature so that we can create the highest yield. So currently, only PCR was successful, and then the other steps were delayed. So I currently can't really answer the question did it work? But during my time here, I did work on other projects. So I did learn how to express proteins, purify proteins. I also learned how to screen crystals, and I did make an SDS-PH gel, along with many other things. So for the conclusion, we're still in the process of discovering a suitable beta-1,3 galactosyltransferase, because currently our genes haven't actually worked. So we can't say yes and we can't say no. But after we make this enzyme, we can hopefully synthesize LNT, and we can add it to infant formula to protect babies from necrotizing enterocolitis. After we make LNT, we can also make derivatives of LNT such as scalylated and fucosylated LNT, and we can hopefully characterize those too. So I would like to thank Dr. Chen and Dr. MacArthur because Dr. Chen allowed me to work in our lab. I would like to thank Dr. MacArthur for being my mentor and being really patient with me. I would like to thank all of the members in the lab for helping me too, and also the National Institute of Health and the National Science Foundation for funding this, the UC Davis School of Education, the UC Davis Department of Chemistry, and of course the Young Scholars Program. So these are my words cited, and any questions? So HMOs, they're consisted of glucose, galactose, N-acetylglucosamine, and N-acetylgalactosamine. So by making by attaching different ones of these to LNT, we can create different derivatives. And so all these different combinations that can be added onto the ends of LNT can create different modified versions of LNT with different characteristics. Oh yeah, so to make LNT, previous research was successful in making LNT, but the yields were too low to be applicable to the real world. They had about a 60 or 70% yield, which wasn't high enough to be added to baby formula and sold at a reasonable price. Next up is Halal Morar.
Yeah, we're really early, so we're going to wait a second. Yes.
Hello and welcome everybody. My name is Paul Morar, and here at UC Davis I've been working under Dr. Dinesh Kumar in the Department of Plant Biology to research how plant pathogens evade plant defenses. So just to clear up any confusion, plant defense does not look like this. Instead they defend themselves on a molecular level. So currently we have about 7.5 billion people in the world, and by 2035 we're expected to hit 9 billion. And there's this problem with the increasing rate of population, and that is that we cannot sustain this many people in the world while still losing food crops. We don't get 100% of the crops that we grow, and we don't get anywhere near 100%. So in order to address this, we need to develop pathogen-resistant plants. So we lose food crops due to nature, bugs, and 10-15% just to pathogen infections. So developing those pathogen-resistant plants would be crucial to reduce this percentage and get more crops onto our dinner table. So plants defend themselves similar to how our bodies do. They have immune systems, and just like we do, and no matter how weak or strong they are, they have two main defense systems. The first one is called pattern-triggered immunity, which relies on the perception of a pathogen molecular pattern on the outside of the cell. Once that has been detected, it then triggers the initial immune response. But pathogens have evolved over the years and inject these effector proteins into the cell. These proteins go to inhibit those initial receptors on the outside and take out the cell first line of defense. So now the cell is more susceptible and more vulnerable to an infection. And these effector proteins enter the plant cell through the peptide secretion system. And what happens here is that an effector travels through this transmembrane base on the bacteria cell, through a long needle, and then injected directly into the plant cell. But the question is, where do these effectors go once they are inside the cell? Research has shown that the effector localizes to the plasma membrane. And by seeing that they localize to the plasma membrane, we can infer that they are trying to take out those first receptors on the outside, making the cell more susceptible and inhibit those receptors, leading to the triggering of the second immune response, which would be a lot more effective, usually leading to cell death. So where else do these pathogen effector proteins go? Do they go to more than just the plasma membrane once inside the cell? So in order to address this, I use the technique of molecular cloning. This consists of PCR, which is the amplification of a gene billions of times, a gene of interest, and that PCR product then undergoes isothermal assembly. This is also called molecular stitching, and what happens here is that a DNA plasmid is cut open at a very specific site, and your gene of interest is inserted into this plasmid. On the bottom figure, I show that where the effector goes into the plasmid of interest, is then meets with GFP gene, where the two are then fused together and join the rest of the plasmid sealed and stitched together. The gene of interest is tagged as GFP to assist in localization and visualization of where these effectors go once inside the cell. So this plasmid is then transformed into competent E. coli cells. These cells take in the plasmid and begin to naturally replicate and increase the concentration of DNA. Once we have that, we then perform mini prep on those bacteria and purify the cell free from all other organelles except the DNA. So now we end up with just purified DNA. This purified DNA sequence is then sent off for sequencing to a sequencing company, where we get back the results electronically. We confirm the sequence to make sure that it codes for a protein of interest, rather than coding for nothing at all, or a protein that is not of interest. We then prepare, once we confirm the sequencing, we then prepare for infiltration. We use agrobacteria to prepare for infiltration because agrobacteria are naturally efficient and effective at transferring their own DNA into a plant cell. So once the agrobacteria have our DNA of interest inside, it then transfers that into the plant cell, where it goes directly to the plant cell genome. This is what the plants look like, the tobacco plants look like before infiltration. We see on both images that each leaf is large, healthy, very green, and free from any blemishes. And these pictures were taken 4 days after infiltration or exposure to the pathogen. And we see in the red boxes that where this pathogen has made contact with the cell, the leaf, is where the cell has withered and died. So this leads to the plant leaf appearing to be infected, which it is, and a lot of cell death. But the sites where the pathogen did not encounter are still very green and healthy. A sample was taken from each of these plant leaves and then underwent microscopy. In the microscopy, we can visualize the GFP and where it went in the cells, since the GFP is attached to our effector protein. And on the right, we see the magenta shade being our indicator for chlorophyll A. Chlorophyll is found inside the chloroplasts, so the right image, or magenta shades, are just to help us show where the chloroplasts are, since they are naturally green. So specifically for effector 14, we see that it localizes to the plasma membrane. This is shown with the GFP fluorescing from the plasma membrane, and we can confirm that these are not chloroplasts, since the pattern on the right and the left do not match. So wherever we see the chloroplasts is not where we see the GFP glowing, and that's where the effector has gone to. So effector 14 has localized to the plasma membrane. In effector 15, we see that there has been an unequal distribution in the GFP. So it is not as clearly distributed as effector 14. So here we can infer that they are targeting lipid rafts. And what lipid rafts are these regions in the plasmid membrane bilayer where there is an extra concentration of lipids and proteins. And we can confirm that this GFP fluorescence is not from chloroplasts, since there is little to no magenta fluorescence on the right image. In effector 17, we see that this is the first sample where we got and saw that it localizes to chloroplasts instead of our expected plasma membrane results. So here we see that they do localize to chloroplasts, since the patterns on the left and the right are identical. So wherever there was a chloroplast, there was a GFP fluorescence. In effector 22, this was the same results as 17, where it localized to the plasma membrane. This is again shown with the same patterns on the left and the right, symbolizing that the GFP has localized to the chloroplasts. In effector 24, we see that they have localized to mesophyll cells. So now we added a third organelle that it localizes to. We have plasma membrane, chloroplast, and now mesophyll cells. But what's interesting about effector 24 is that it only localized to the membrane of the mesophyll cell. So we can see in the right image that we see structures inside the mesophyll cells, but we don't see GFP on the left side inside the cell. So we can say that it's only localizing to the mesophyll cell membrane. This is another image of effector 24. This was taken with a bright field. So we can see the GFP glowing, fluorescing from the plasma membrane. So what was interesting about 24 is that in both of these pictures, is that this one localized to mesophyll cell membrane, and this one localized to plasma membrane, the cell plasma membrane. So we generalized the results to be that the effector 24 localizes to membranes in general, not specific to the plasma membrane, and not specific to any other membrane. It just targets membranes in general. So this research has shown that there has been effector localization to plasma membranes, but this has also shown that chloroplasts and mesophyll cells are also potential targets for pathogen effectors. Pathogen effectors target these organelles because they could be crucial to plant cell defense and immune responses. So the effector protein and the pathogen would want to inhibit these organelles where it could begin or trigger another immune response. So we could further investigate what the chloroplast and mesophyll cells do to contribute to triggering an immune response. And we could also investigate which region specifically that these organelles target, that these effectors target inside the organelle. Because organelles are comprised of many different types of components and parts, it could be targeting a specific region of these organelles rather than just the organelle itself. So investigating these two things could lead to to the development of a solution to fixing these pathogen-infected crops so we can get more food on our table rather than losing them to these pathogens, since we have this increasing population and we need to find a solution to this quickly. I would like to thank Dr. Dinesh Kumar for allowing me to do research in his lab and giving me this wonderful opportunity. I would like to thank Neeraj Lal, who's a graduate student who guided and mentored me and explained concepts over and over again with me and took a lot of time and patience with me to walk me through a lot of what we did. I would like to thank Gita and Cindy, who are undergrad students, who also helped me and guided me and answered my questions throughout lab. And I would also like to thank Carla for addressing my questions and concerns about the program. Are there any questions?
Why does effector triggered immunity come after pattern-triggered immunity?
So the question was, why does effector-triggered immunity come after plant-triggered or pattern-triggered immunity? So what happens here is that the pathogen injects these effectors after it's been detected. So once these effectors are injected, it then begins to encounter the effector-triggered immunity. And this comes after pattern-triggered because pathogens have evolved to evade this initial plant defense of the pattern-triggered immunity and actually get into the cell before being stopped or killed off by the cell immune response.
Are there any other bacterial species that can transfect plant cells, such as the agrobacteria, besides the agrobacteria?
So we studied pathogens from three different. Pathogens from three different types of pathogens. We studied nematodes. We studied bacteria and fungi pathogens and see their effect on plant exposure and see what's the interaction between the pathogen and the plant.
How long does it take for the signs of infection to show up on the pathogen-infected leaf?
So we begin to see a result of the infection after 2 days. So if we infiltrate on Monday, we would expect to see results on Wednesday and see that there has been infection occurring with black specks on the leaf, or just what we saw earlier with the most of the leaf being dead since the effector-triggered immunity triggered cell death to prevent the pathogen from spreading. So we see those results in about 2 days.
Why are lipid rafts potential targets?
So lipid rafts are potential targets since there's a high concentration of lipids and proteins. These proteins can be those receptors from the pattern-triggered immunity, so it's targeting those receptors specifically. And it could also be a method of cell communication, where the infected cell can send signals to other cells and say that there's an infection and you need to prepare yourself.
So how does knowing about the localization help aid in the development of the resistant plants you're talking about?
So knowing where these effectors go in the cell and what they target can lead to the development of a better supplement or antibiotic to help these organelles strengthen their defense or their membrane, so that way the pathogen would have a more difficult time infiltrating and getting into the cell or a specific organelle. So this way we can develop a better supplement for these organelles.
So earlier in the presentation, we were talking about infecting the tobacco plant. How did you infect it?
So the question was, how did we infect the tobacco plant? And what we did was that after we got our agrobacteria transformed cells, that DNA was then injected into plant cells. But before that, we prepared a cell culture of those agrobacteria, and that was injected by scraping a little hole onto, not a hole, but just scraping the surface of the leaf, and then using a needleless syringe to inject that into the leaf. And you could see the effector solution spread across the cell, the plant leaf, once you scraped and injected.
Why did you choose a tobacco plant rather than any other?
That's a very good question. So we use tobacco plants since they have been established with previous research done long before. So we know about their how long they take to grow, about their defense systems, about a whole bunch of other things that happen inside the cell. So and we use that since it's already been established rather than establishing a new plant as the researching plant for these for plant defense, because that'll just take a lot of time to determine all the aspects of the plant, including the growth and the defense and how it reacts to different stimuli. Thank you, and next up we have Brianna and Cheryl.
Hi everyone, my name is Cheryl Young.
And my name is Brianna Lee.
And our project is on sunflowers, specifically heliotropism, auxin, and circadian clock. We worked under the direct supervision of Dr. Hakovatamian, postdoctoral researcher Hakovatamian, in Professor Harmer's lab in the Department of Plant Biology.
So phototropism is the directional growth of a plant towards a light source, as you can see here. Heliotropism, on the other hand, is the dynamic form of phototropism. In the morning, plants face east and track the sun throughout the day towards the west. Interestingly, at night, they reorient to face back towards the east in anticipation of the sunrise.
So how does the plant move to face the east and the west? So throughout the day, the east side of the plant grows faster, which causes the plant to face west at dusk. At night, the opposite occurs. So the west side of the plant grows faster, which ultimately causes the plant to face east at dawn.
So besides the fact that solar tracking, or heliotropism, is aesthetically pleasing, it's also important for optimal growth. The Harmer lab tested this by tethering half of their experimental plants, as you can see here, and left the other half free to move. They found that the tethered plants, represented here by the blue bar, had a lower biomass than the control, represented here by the red bar, and also had a smaller average leaf area, as you can see here, in comparison to the control, as you can see here.
Therefore, heliotropism causes these plants to grow larger. This information is important because scientists may be able to increase the productivity of other economically important crops.
So auxin is an important plant hormone that's involved in many different plant physiological processes, such as phototropism, which, if you can recall, is the directional growth of a plant towards a light source. Auxin, represented by these pink dots, naturally move away from a light source, causing the cells in the shaded side of the plant to grow faster and cause cell elongation, which ultimately pushes the plant towards the source of the light.
So previously, Professor Harmer's lab also showed that the circadian clock plays a role in solar tracking. The circadian clock is a biological timekeeper that causes organisms, including plants and animals, to measure time. Using the circadian clock, these plants and animals will change their physiology to match their environment. Other processes regulated by the circadian clock include jet lag in humans, leaf movement, and flower opening and closure in plants.
So for my part of the project, we hypothesized that auxin plays an important role in sunflower heliotropism. We quantified heliotropism by drawing dots on the plants and measuring the angle from the tip of the plant with respect to the horizon line. At dawn, the angle was less than 90 degrees, which indicated that the plant was facing east. At dusk, the angle was greater than 90 degrees, which indicated that the plant was facing west. we plotted these angles on a graph. And so in the morning, when the sunflowers were facing the east, they had a lower angle of curvature. As they tracked the sun throughout the day, their angle of curvature increased, indicating that they were facing west. At dusk, the sunflowers reoriented back towards the east, as you can see by the decreasing angle of curvature. So we applied auxin by first pouring a controlled treatment or solution into a lanolin paste, which is a sheep fat. We then mixed the solution into the lanolin and applied it on the appropriate side of the sunflower. We applied auxin in a combination of 4 ways. We applied it on the east side at dawn, shown here, on the east side at dawn, shown here, on the east side at dusk, shown here, and on the west side at dusk, shown there. We recorded our data by taking pictures using this camera every hour. We checked for the appropriate settings, and we mounted the camera on a stake. We ensured that all of our plants that we were experimenting on were in the frame, usually around 2 to 4, and then we tied the camera to the stake and checked the images for clarity. So in the field, we drew dots on our plants so that we could measure our angles using this method, if you remember. We used the program ImageJ to record the angle of the plant every hour for 48 hours and plotted those angles on a graph on the y-axis with the x-axis as time to create figures like this. So last season, experiments were done using 2,4-D, a synthetic type of auxin, that suggested a role of auxin in solar tracking. This season, I used IAA, a natural form of auxin, to see if it would have the same results on heliotropism. So the first experiment that we conducted was applying IAA on the west side at dawn. Auxin is naturally found on the east side so that it can help push the plant towards the west throughout the day. So we predicted that the applied auxin would inhibit that westward movement throughout the day. The first 24 hours were used as a control. As you can see here, the control plants and the treatment plants followed the same track, following the sun throughout the day from east to west, and then reorienting back east at night. When the auxin was applied here, the controlled plants followed the same path, but the treatment's westward movement was inhibited, as you can see, by the lower angle of curvature. Our results matched last year's results. As you can see, both treatments have a lower angle of curvature than the control. So for our second experiment, we applied IAA on the east side of dawn. This is the same side that auxin is naturally found in, so we expected an enhancing effect towards the west throughout the day. Again, the first 24 hours were used as a control, and when auxin was applied here, the treatment's angle of curvature was bigger than the control, which indicated that its westward movement was enhanced as we expected. Our results showed similar patterns to last season's results, but more repetition is necessary to confirm this year's results. So for our third experiment, we applied IAA on the east side at dusk. Auxin is naturally found on the west side at this time, so we expected an inhibition of movement towards the east at night. When auxin was applied here, surprisingly, there were no effects. There was no inhibition as we expected, and so when the plant switched directions in the morning, the IAA that was applied that we expected to inhibit at night had an enhancing effect in the morning, as we can see here by the greater angle of curvature than the control. This season's results matched last season's results, as you can see here by the increase in the angle of curvature for both of the treatment plants in comparison to the control. Finally, our last experiment was applying IAA on the west side at dusk. This is the same side that auxin is naturally found in, so we expected an enhancing movement towards the east throughout the night. Again, surprisingly, when auxin was applied here, there was little to no effect at night. However, when the plant switched directions in the morning, the auxin that we applied that we expected to enhance its movement caused an inhibition effect, as you can see here, by the lower angle of curvature than the control. Our results showed similar patterns to last season's results, but again, more repetition is necessary to confirm this year's results. Therefore, my hypothesis that auxin plays an important role in sunflower heliotropism was rejected. My new hypothesis is that auxin plays an important role in sunflower heliotropism only during the day because there were no effects at night.
So previously, the Harmer lab indirectly identified the role of the circadian clock in solar tracking. Right now, for my project, I am directly studying the role of the circadian clock on the solar tracking based on a molecular level. So the circadian clock has three parts. The first is input pathways, the second is central oscillator, and the third is the output pathways. So the circadian clock uses the inputs, such as light and temperature, to adjust or synchronize the central oscillator or the internal biological clock to match the external time of the day, producing rhythmic outputs such as sleep-wake cycles and knowing when to be awake and knowing when to be asleep. The circadian clock has one of the main characteristics is that it can continue producing these rhythmic outputs even in the absence of the inputs, just like in jet lag. So say you're traveling from California to India. The time difference is approximately 12 hours, or half of a day. So when it's 8 p.m. in California, it's really 8 a.m. in India when it's light out. So naturally, during the day in India, your body would want to sleep. And this is because your body is adjusted and trained to the California time. And it's ignoring the Indian time until it realizes that the inputs in India are persistent and it adjusts your circadian clock to the Indian time. So the circadian clock does not look like an actual clock, and nor does it run on batteries. Instead, in plants, specifically in Arabidopsis, it's made up of a network of genes that control and regulate each other at specific times of the day. Previously, the Harmer lab showed that the circadian clock plays a role in solar tracking. So remember that the circadian clock can continue producing rhythmic outputs even in the absence of inputs. So in this figure, they had plots of sunflowers planted in the field for 2 days in natural conditions. So they followed the solar tracking behavior as Brianna explained earlier, where they moved towards the west during the day and moved back towards the east at night. However, on the third day, they moved the plants indoors so that they were in a constant light chamber for 48 hours. As you can see, they continued their heliotropic movements for several days, meaning that the circadian clock may play a role in this solar tracking. So what I wanted to do was disrupt the circadian clock and to see its effects on the solar tracking. Specifically, I disrupted the Lux gene right here, which is part of the evening complex, which previously has been shown to disrupt the entire circadian clock in Arabidopsis when disrupted. So my hypothesis is that disruption of the Lux gene will disrupt the entire circadian clock, which will then disrupt solar tracking. So some of the genetics behind my project. I'm looking at the DNA sequences in the Lux gene. A gene is essentially just a region of DNA sequences on the DNA, and that is then expressed to form proteins. Sunflowers are diploid organisms, meaning that they have 2 sets of chromosomes, and thus they have 2 forms of the genes called alleles. And they receive 1 set of chromosomes from their father and 1 set from their mother. So let's say that our parents are both heterozygous, meaning that they have 1 form of the allele and then another form of the allele, here big A and little a. So when we form a Punnett square, we will have 3 different genotypes. We will have big A, which is homozygous dominant, big A, and then little a, big A, which is both heterozygous, and the last one being little a, which is homozygous recessive. So our genotypic ratio is 1 to 2 to 1, and our phenotypic ratio is 3 to 1, since the heterozygous and the homozygous dominant forms exhibit the same phenotype. In my project, I was looking at the homozygous recessive genotype because that is what shows the Lux mutation phenotype. So my overall procedure was to amplify the region around the mutation shown here in red. And it's simply just a base pair change. And then I used a restriction enzyme that cuts the non-mutated gene. However, it does not cut the mutated gene. So my first step in this process was to perform DNA extraction. I planted 20 mutant sunflower plants that were collected from, the seeds were collected from a heterozygous mutant sunflower head. And then I performed DNA extraction by grinding up the frozen leaf tissue in a mini bead beater here. And then I performed a series of steps to precipitate out the DNA and to wash the DNA with alcohol. And then I could finally test the concentration and quality of the DNA. After I had extracted DNA, I could perform PCR, or polymerase chain reaction, which has 3 crucial steps. The first of which is melting, so the DNA strands break apart. The next step is annealing, where the primers attach onto the DNA templates. And the third of which being is elongation, where Taq polymerase will extend the DNA out. So this process, or chain reaction, repeats itself in cycles for many, many times until we have millions and millions of copies of the region around the mutation. So in order to visualize my amplified PCR product, I performed gel electrophoresis. Gel electrophoresis essentially has a negative side and a positive side, where the negatively charged DNA will migrate towards the positive side, and smaller pieces of DNA will move faster and move further, while longer pieces of DNA will move slowly and travel less distance. So this is a picture of one of my amplified PCR products. As you can see here, the top bands are basically almost all of them are amplified, and the last one over there is the control, while these bottom bands are the primers that were used in PCR. So now that I had my amplified PCR product, I was ready to perform restriction digestion. So restriction digestion is essentially cutting up DNA molecules into smaller pieces based on specific sequences. They do that through restriction endonucleases. And the way that restriction endonucleases work is they scan the DNA, then they recognize the specific sequence, and once they recognize it, they will cut or cleave that DNA at that specific spot. So pretending that this is a non-mutated gene, and we found that there is an enzyme called BSTU1 that cuts this non-mutated gene. However, the BSTU1 does not cut the mutated gene that has the single base pair change. So instead, it only scans the DNA, does not recognize it, and cannot bind to the gene. So in order to visualize my restriction digestion, I also performed gel electrophoresis. And with all these different combinations of alleles, what do we expect to see on our gel? So if we have 2 normal alleles, 2 normal forms of the gene, we expect to see 2 bands, because BSTU1 cuts the normal allele. If we have 2 mutated alleles, meaning the other form of the gene, we should only see 1 band, because BSTU1 does not cut the mutated allele at all. And if we're heterozygous and we have 1 normal and 1 mutated, we should see 3 bands. So one of them would not be cut, like in this case, and the other allele would be cut, like in the normal allele. So these are my results of the restriction digestion. And more specifically, if we take a look here, this right one is the undigested one, meaning that we didn't add a restriction enzyme to it. And here we see that there are 3 bands. In the other case, meaning that's heterozygous. In the other case, this one also undigested, meaning that there's no restriction enzyme, there are only 2 bands, meaning that it's homozygous dominated. So our results, our expected results based on the Mendelian ratio of 1 to 2 to 1, we should expect to see 5 plants that are homozygous dominant, 10 that are heterozygous, and 5 that are homozygous recessive. However, in my experiment, we had 5 homozygous dominant plants, 15 heterozygous plants, and 0 of the homozygous recessive plants. So this does not match the Mendelian ratio, and we believe that there are possibly 2 reasons for this. One of which is being that we had a small sample size, relatively small, only 20 plants, and because the ratio is based on probability, and there's a lot of variation in smaller sample sizes. The second theory, although highly unlikely, is that it's possible that the Lux recessive mutation, that the 2 recessive alleles, is lethal, meaning that if the plant had the homozygous recessive mutation, the plant would die, and we would not even be able to see it. So future work will of course first start with redoing this entire process to find a recessive mutated plant. And then we can plant it in the field and observe any phenotypic differences from the typical solar tracking behavior. And based on that, we can either accept or reject the hypothesis. And using that information, we can apply that to other economically important crops in the agricultural industry, because as sunflowers track the sun as a way to adapt the environment. So knowing how sunflowers track the sun, we may be able to apply that information to other crops as well. So we would like to thank Dr. Hagamatanian for being our mentor and guiding us through both of our projects, as well as the entire Harmer Lab for welcoming us and allowing us to do research.
We also appreciate the Young Scholars Program and Dr. Rick Pomeroy for giving us the opportunity to perform research this summer, as well as Carla Sison, who has helped us through the entire experience, and all the other counselors and students. Are there any questions?
So no, it's only when the young plants that they'll be solar tracking. As soon as they become flowers, they'll stop tracking the sun. So like when you go to the grocery store and you look at the sunflowers, they're all facing a certain direction.
And they end up facing east because in a previous experiment they found that these tend to grow to pollinate the flowers that face east and not the ones that face west.
Chelsea?
Why was the research enzyme unable to cut the mutated gene?
Oh, so what happened is that the base pair change is what removed the restriction site. So then that enzyme could not recognize that DNA sequence. Ria?
Could you explain the purpose of the lamellin area? Like what did you use the sheet back for?
So basically we just used it to apply the IAA to, so we had a solution and we poured it into the lanolin paste so that it would stick and stay on the plant.
Yeah, because like the plants have a waxy coating, so we didn't want the IAA to, like drip off or evaporate. Hello?
What was the gene or allele that was being talked about in the homozygous recessive?
It's the Lux gene. Well, it's the Lux gene in Arabidopsis. It's fugitively a Lux gene in sunflowers. So it's part of the evening complex that represses day phase genes. So it represses the expression of the genes in the day.
What's the process of growing a plant that you guys like to use?
What's the what?
What's the process for like growing a plant, and like how long does it take to get to the point where you can take it out and look at it?
Well, okay, so you mean just like the growing the plant?
Yeah, like in the lab.
So so we planted them in like trays of soil, and we put the seeds on, and then we like lightly covered it with the soil so that they would all germinate at the same time, and they would be like equal growth rates. And then after approximately, I think germination takes about
It's about a day.
or a couple of days, and then after about a week so basically we extracted the DNA from the true leaves. So when the seeds first germinate, they have these two, they look like leaves, but they're not leaves, they're called cotyledons, and they're part of the seed. And then we had to wait for the true leaves to sprout up for us to do DNA extraction on them. So that took about, I think, a week for us to get the true leaves. And then, yeah, so we extracted the DNA from smaller plants. It didn't have to be huge plants. Archita?
You mentioned something about the homozygous recessive and it was the Lux gene being lethal.
Yeah.
Did you know how to be able to tell if it was really quick?
Yeah, it's highly unlikely since the Lux gene is just a gene in the circadian clock. Like, it's not necessary for the plant to function, like if it was a gene that regulates photosynthesis or something like that. I guess the way that we would be able to tell is if we continued more trials of this and we noticed that none of it was popping up. Since I only did 20 plants here, we think it's more due to the probability. But if we did more and it was always no homozygous recessive, we could probably say that it's possibly due to that lethal.